Good evening, everyone, fellows, partners, permanent fellows, guests, friends of the Wissenschaftskolleg. Welcome to our evening lecture tonight, and a very special welcome to the members of our academic advisory board, who will have their selection meeting tomorrow. And I take the opportunity to express our deepest gratitude for all the effort and time you invest in selecting a group of fellows every year, an amazing group of fellows, and without you, the WICO wouldn't be what it is. And I would like to especially welcome 3 new members of the advisory board. Former fellow Judy Bronstein, there she is. Also a biologist, and also former fellow Insa Nolte, African Studies, and again, former fellow Eric Monet, economist. There you see. Over there. So, but this is a very special evening also for another reason, namely Dieter Ebert, distinguished evolutionary biologist from Basel and our permanent fellow for 10 years, and our very dear advisor in all matters of life sciences, will give his farewell lecture tonight because his second term as a permanent fellow has come to an end. So a particularly warm welcome to our guest of honor, Dieter Ebert. And you see that this is a very special evening also for Dieter, because this is the very first time that I see him wearing a suit and a tie. And if you look at the tie more closely, as he explained to me, it shows hosts and parasites. So have a look at the tie. Dieter Ebert contributed significantly to shaping not only, of course, the academic profile of this institute, but also the social atmosphere of the Wissenschaftskolleg. And he did so for at least 4 significant reasons, or because of at least 4 features, namely his academic competence, of course, his extraordinary commitment to this institution, his talent to fascinate non-experts in the field of biology, and last not least, for certain extra academic skills. And let me elaborate this a little bit, these 4 points a little bit more. First and foremost, of course, his excellent academic profile. Dieter, as I said, holds the chair of zoology and evolutionary biology at the University of Basel since 2004. After receiving his PhD also in Basel, he gained a lot of international experience, from Oxford to Ascot, through the Russian Academy of Sciences in Irkutsk, as I learned from his CV, to the Smithsonian Institution in Panama, and finally, after a brief interlude in Fribourg in Switzerland, he returned to Basel again. I won't enumerate all his memberships and honors and so on. I only mention that he is a member of the German National Academy Leopoldina and a distinguished fellow of the European Society for Evolutionary Biology. His main field of research is the relationship, as I said, between hosts and parasites, their interaction, their ecology, their genetics, their corresponding evolution. So a dynamic antagonistic relationship that can be traced back millions of years, which is also the topic of his talk tonight. And studying this is extremely important if we try to understand, for example, the dynamics of infectious diseases and many other things. His favorite model organism for studying this is Daphnia magna, or, as I learned, the water flea. Actually not a flea, but a crustacean, a modest species measuring just a few millimeters in size, if I'm correct. And in Basel, as I was told, he has gathered a unique collection of all kinds of water fleas from more than 4 continents and 40 countries. What makes Daphnia magna so apt as a model organism? Daphnia can be found in almost every pond, lake, or even puddle, And they are frequently infected with diverse microparasites, so you can analyze the impact of parasites not only on individuals but also on entire populations. And then you can easily replicate the experiments and get significant numbers for statistical insights. And what is particularly special about Daphnia magna is that they are able to reproduce both sexually and by parthenogenesis, so that genetic factors can be distinguished experimentally from environmental factors. Very, very special kind of species, obviously. And as you can imagine, the rapid progress of DNA sequencing and big data processing has revolutionized Dieter Ebert's research, like so many others, since his core questions cannot be answered without going to the genetic level. But he is still pursuing classic field work as well, such as observational studies in Finland, for example, but also in South Africa, where he just spent a year as a fellow of the Stellenbosch Institute for Advanced Study. Second point, Dieter Ebert's extraordinary commitment to the Wissenschaftskolleg. Studying your CV, Dieter, as I told you, I found out that your career almost took off here, the Wissenschaftskolleg. One of the very first, maybe the second, international workshop you attended generally, as a very young scientist, took place here in Vico in 1993. You later became a regular Vico fellow in 2010-11, when you were invited as a member of a multidisciplinary focus group on infectious diseases. And Vico obviously did not want to part with you, so you were elected, a few years later, you were elected in 2015 permanent fellow. Dieter's 10 years service as a permanent fellow and as a member of various selection committees contributed significantly, as I said, to shaping this institution. Thanks to you, Dieter, I think Vico has won over many of the most stimulating evolutionary biologists in the world, like some of the current fellows too. Over the last decade, I would say hardly anyone else promoted life sciences at Vico as successfully as Dieter did. He put Grunewald, I think, on the mental map of biologists all over the world. And hardly anyone else has been so committed to the strategic development of our selection procedures and the general character of this institute. The fact that he has strengthened the life sciences here so much is third point, due to his talent and willingness to explain complex research topics in a clear and simple but not simplistic way. And it's always been a pleasure, an intellectual pleasure, to listen to him explaining to us non-biologists, non-life scientists, what biologists do and why we should care. His didactic enthusiasm, by the way, also explains why he has attracted so many talented graduate students over the years who have gone on to scientific institutions all around the world, from Australia to the US to Japan and Brazil. Fourthly, when talking about Dieter's merits, I also have to mention his contribution to the more mundane dimensions of Vico. He comes from the Mosel region, close to Trier, which is, as you all know, an old wine-growing region, ancient wine-growing region. And this also explains, I think, his enthusiasm for Stellenbosch, which is also a wine. And his brother has a vineyard from which we at the Vico benefited a lot. And the fellows won't forget the wine tasting that Dieter organized 2 years ago, and I won't go into the details here. So to conclude, on behalf of the whole Vico, I'd like to express our gratitude for a long, fruitful, very collegial, and also really pleasant relationship. The end of your permanent fellowship really marks, I think, a caesura for this institute. But we are not quite there yet. Our relationship is not about to end today. Dieter, together with our current fellows Leonie Moyle and Matthew Hahn, are in charge of an observatory, so-called observatory conference, titled Revolutions in Evolution. That will take place on February 25th to 27th, starting with a public panel discussion on the evening of February 25th. So save the date already. That will be, I think, a very interesting evening. So now, can the present explain the past? As a historian, I would say, of course, it can. But you'll be talking about a 15 million year long past. And I'm curious what you will have to say. So without further ado, the floor is yours.
Oof. Thank you, Barbara, for this really very friendly and generous introduction. You said about everything about Daphne already. I actually can stop here, you know. We can continue with the wine. Okay, dear fellows, permanent fellows, current fellows, friends, colleagues, guests, it's really a very big pleasure for me to stand here in front of you and talk about work which my group and myself have been doing over the last 10, 15 years, which I try to summarize in a, how did you say, simple but not simplified way. And I hope you will all have some fun with this. So what you can see here is a landscape or a waterscape. It's a small pond, and you see there are plankton organisms. These are water fleas. But you see there are not only water fleas, you see also there are other organisms. Sometimes you see insects rushing through there. These are predators. Sometimes you see little beige points going through the water. These are ostracods. Here's one coming in the center. And all of them have antagonistic relationships. So this is, it looks a very peaceful and harmonic picture. There's actually a constant war going on here. This insect trying to eat the Daphnia. This little thing what just went through here eats the eggs of the Daphnia. And if you look, quite a lot of the Daphnia are whitish inside. They are parasitized. They carry a parasite inside which changes the color. The world is a dangerous place. And this is not only now, this has been like this for a long, long time. This is here a picture from the Cambrium, 500 million years old predator, a parasite from the same time period. And of course, when you talk about danger, you can't leave out T-Rex, who was 70 million years ago eating other dinosaurs. Possibly, this has been like this for the last 4 billion years, ever since life began. Antagonistic relationships, host-parasite interactions, is arguable, no, how do you say it? Well, you can argue about it. The oldest form of selection you have in natural habitats. It's everywhere, possibly soon after the first forms of life on this planet came about, the first parasites were there and exploited this, made use out of it to do their own business. Meaning they evolve to extract resources from the host, they increase the survival, they increase their reproductive mode, and they foster their transmission to other hosts. And all of this at the cost of the hosts, which are suffering, dying, all types of problems, morbidity. And this has been an important event in evolutionary history in general, but also in human history. Think about the Black Death in the Middle Age, or possibly, I don't need to remember you, what happened here 5 years ago. This chap here changed the way how humans interacted worldwide. But hosts are not only victims, hosts also fight back. So we have a a lot of mechanisms evolved to help us, to protect us against parasites. This is just kind of a cartoon showing the different ways how we are trying to cope with parasites, trying to reduce the likelihood to become infected or trying to reduce the damage done by parasites. So taking these two things together, what we will have is a scenario where the parasites increase constantly to damage the host for their own means, and hosts try to escape this. And out of this results a scenario which we call coevolution. Coevolution is the reciprocal influence of two populations, in this case antagonistic population, but this can also be a mutualistic interaction when they benefit each other, but here I'm talking only about antagonistic relationship. Now when I started to become interested in this, which was about 30 years ago, I talked with people about this and they were telling me kind of their ideas how coevolution works, and I found all of this very fuzzy. Looking into the literature, it turned out there was actually almost no clear concept how coevolution would work. Everybody liked to talk about it. Everybody had this idea of hosts evolving, parasites evolving, and so on. But how it works was unknown. And I decided, okay, here's a niche, and this is something we need to know because it's so important. So I decided I want to move into this and try to understand how coevolution actually does work and what it is doing. Now, as I said, at this time there were no clear concepts about, but concepts emerged around this time. And there were a lot of different ideas people had how it would go. And over time we realized that these concepts fall into kind of two categories. And I want to present those to you now. I start with this one here. So imagine you have a policeman with a club and a gangster with a pistol. That's not very good for the policeman. It seems not that he has an upper hand here. So maybe he should also have a pistol. But then the gangster might come up with a bigger pistol or a more modern pistol. And what you see here already is you replace one weapon with another. And the later weapon is better than the previous one, which means that the previous weapon is losing its value. Obviously, at the moment the policeman has a pistol, the club is of no use anymore. So you replace the previous with a new one. It's selection for the better. And this will continue like this, and possibly even longer. That's a type of arms race which we have. Those of you who are about my age may still remember this. The time of the superpowers developing always new type of weapons, longer reaching missiles, stronger potential, killing more and potentially killing more and more people. This is the newer, the better, and it replaces the previous ones. So the old one loses the value when the new one is coming. But there's a different form of coevolution, which I want to try to explain you with this one here. Imagine you have specific interactions like you have it when you have a lock and a key. The lock can open the key, the key can open, the key can open the lock, and in this case here the yellow key can open the yellow lock and the black key can open the black lock. And obviously the yellow key cannot open the black lock and the other way around. If you now imagine you have a village where everybody has a black lock and there are no gangsters, that's fine, you can lock it. Now a gangster comes with a black key, they can open the door. That's not good for the people in the village, so they try to be inventive. They bring up a new version of a lock, they come up with a yellow lock, and now this black key cannot open this anymore, so the people with the yellow lock are protected. But eventually, the gangsters have a mutation which produces a new key. The new key is yellow, and the new key can open this yellow here. This means that before we lost the value for the black one, and now we had the value for the yellow one, but with the new key coming in here, the yellow one is also losing its value. interesting is that the black one is gaining against value because people focused on the yellow one, and the gangsters with the black key went out of business before. So the black lock has a meaning again. We call this negative frequency dependence. This is a complicated thing. I try to explain you with this graph here. These are data which I collected from the Norwegian Statistics Centralbureau, which is a place where all the data of all names of boys and girls are shown. I took the one for the boys. And you see it goes very far back in time. This is why it shows it. So we go here 1880 until 2020. So we have a long time period. And what you can see here is the up and down in the popularity of different names. So we have here Ole being very popular about 140 years ago. But after some stage, the parents see there are lots of kids walking around all being called Ole. And of course, when your wife is pregnant, you will not recommend her to call the kid Ole. You will look for a different name. What you're looking for is a rare name. So you give an advantage for rareness. And apparently Olaf was very rare at the time, and so Olaf suddenly became very popular. So popular that it replaced Ole, but also after a few years there were too many little Olafs running around. And the family said, no Olaf anymore, let's go for Arne. And so they went for Arne, and then afterwards for Jan, for Christian, and so on. So you have this fashion over time, and the fashion is driven by a time lag that you give the names, and then it takes a little while until people realize, oops, there are too many Oles now around, and only then they are reacting. This is kind of what makes these curves not kind of pointy, but kind of hump-shaped. A second thing what is important here is, you see, for example, Ole was very popular here, and then it kind of drastically went down, and then suddenly, 40 years later, it comes back again. It doesn't reach too high values, but nevertheless, it reaches quite some values. So you recycle an old name kind of a century later. The same here with Christian. Christian was, the blue here, was quite frequent in this period here. And then about 100 years later, it suddenly comes back, and this time even reaches the rank #1 position for nearly 20 years, and then falls down after Matthias and Williams take over. This is time-lagged negative frequency dependent selection. It's negative frequency because rareness gives an advantage, while being common with a time lag is of disadvantage. So it goes down again. These type of dynamics, so you can see here already that there are kind of cycles, it comes back. And you can actually use now this metaphor here with the locks and the keys, and you can think about how would this go over time, and then you get something like this here. So we have the yellow lock here. The yellow lock is kind of becoming quite common because there's hardly anybody out here who can open the yellow lock. They're very rare, the yellow keys. But then the yellow keys have an advantage because there are so many houses around they can open. And then it goes up. And once there are too many gangsters around with yellow keys, the people will not use these locks anymore, and it goes down. And with a time lag, the keys go down as well. So this produces two interesting effects. One is cycles, I mentioned this. The second, what it is doing is, because you never hit the ground here, or you never hit the ceiling here, you're not losing diversity. Both types of locks are always there. And this is important because in contrast to what I told you before about the policeman and the gangster, the new one replaces the old one, and then the old one is out. You don't need it anymore. You don't need the club anymore once the pistol arrived. So you replace something. Here you don't replace it. It stays always there, but at fluctuating frequencies. This model was invented in 1975, and people were laughing about it because they were saying, well, this can't be, this is not working, that's too complex, there's not really anything of importance. The model got the name red-green model. And the reason it got this name is this. You see here this book This is funny. Here we are. This is a book by Lewis Carroll. Lewis Carroll, I'm sure all of you know him because he's the one who wrote the book Alice in Wonderland. So, and there's a second part to this book, Alice in Wonderland, called Through the Looking Glass. And in the second part, there is this red queen, and there is Alice, and you see here this picture where the red queen is running with holding Alice on her hand, and they're running very fast. And then Alice says, well, in our country, you generally get somewhere else if you run very fast for a long time, as we've been doing. The red queen replies, a slow sort of a country. Now here you see, it takes all the running you can do to keep at the same place. And this is, you can think about this arms race metaphor. You're running, 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 but the parasites are running after you. They try to adapt to you and you try to escape their nasty business. And the faster you run, the better, because then you stay on a distance for them, but you cannot stop because in the moment you stop, they will catch you. I come back to this in a second, what happens if you stop. Let's see what happens if you move. This is not real biology in the sense of this is not an experiment. These are observations which were put together to illustrate a point, but it's quite a good illustration, so I want to explain you this. In England, people like to drink beer. For beer, you need barley. And so they breed a lot of barley there. And they have a problem with a fungus, a fungal pathogen. And the fungal pathogen is called mildew, it's over there. And mildew is quite devastating for the field, so you really don't want to have it. And then one day, a variant was introduced, this MLA12 here. And this variant was resistant to mildew. And so it was introduced in 1967. And so the farmer said, oh, wow, this is really great. You see, a year later, 68, we had already 10% of all barley fields with just one clone. 69, 20%, 70, 30%. But oops, what are you seeing suddenly? It becomes infected. You see, here's the infectivity index. It goes up here. And then, of course, once it becomes infected, the value for the farmer declines. And the farmer says, oh, we're not going to plant it anymore. So they take it out. You're moving from here over here. There's one data point missing. Here, and you see we're back down to 10%, 73, we are back down to 3%. But what had happened now? The infectivity went down again. With a time lag, you go back down. And now the farmer said, oh, well, this is only 3%, low infectivity, let's plant it again. 74, 75, 76, 77, we're at 35%. And again, infectivity moves up, we're doing a second cycle. This is the dynamics you get when you have this negative frequency dependent selection. And you always keep all variants on board. Now I said before, what happens if you stop running? If you stop running, this is what our agriculture is doing. You may have heard about the famous monoculture effect, that you have these huge fields, all one clone, all equally sensitive or equally resistant to the pathogen, hopefully equally resistant. But then, of course, because it's such a big target, earlier or later, a pathogen comes which is able to overcome the resistance, and then it has an easy play because everything is there. And they're all the same, and the parasite can spread super rapidly, and then is when we have catastrophic loss of crop because of this consequences of a halted arms race. This was initially thought to be only a problem in agriculture. What is special in agriculture is that plants are sitting in the ground and they are not moving. So this is different from animal populations which are moving around and so on. People thought, okay, this is not a problem there, that's a different scenario. So one of the earlier things we were doing is we tried to test this. Here's the water fleas again. Barbara gave you already a primer what water fleas are. I need hardly to say anything anymore. You can keep them in such containers here. This is about 50 liter each, and in 50 liter you can keep about a population of 1,000. So this is big enough to mimic like what would be one field or one population. And then we introduced this parasite here. It's a fungal parasite which infects this Daphnia. This white stuff here, this is all the parasite. So we created populations which were either monocultures, they had only one single clone of Daphnia, or they had many different clones. So high genetic diversity as you would find it in a natural population. And so we started here without parasites, and then we added the parasite, and then we monitored this over some time. You see, by the end of the first summer, here we have already a much, much higher infection rate in the monoculture as compared to the polyculture. Diversity protects you against parasites. And this continues into the second year here, and the effect stays the same. Diversity matters. Now, so far to the concepts. Now you understood all the background. Now for the rest, it's easy peasy for you. You can follow this without a problem. So, what I want to do now is I want to bring you to a time travel over about 15 million years. So here's our timeline. This is today. There's Corona. These are important dates in history, like foundation, Vico here, University of Basel, pyramids of Gizeh, then Ice Age, and here humans split from chimpanzee. So we have this entire spectrum here. This is how geologists call this. There are different names. And what I want to do with you is to go backwards and show you experiments we have done which relate to these different time periods to understand what form of coevolution is actually happening in these water flea populations. Now, if you do so, you have to be aware that you are kind of you take a different mindset depending on where on the time axis you are. When you are here on this side, this is basically where you see the processes, you can see the dynamics, you have a fine time scale resolution. People might call this microevolutionary pattern, and you have the mindset of a kind of an evolutionary ecologist. Somebody who would like to get its boots muddy, to go out to the field, to look for the water fleas in the pond and all these things. If you go further back in time, you're more and more going on to the observational situation, so you look for patterns. It's more a macroevolutionary perspective you're taking. You're taking a different working approach. This is a lot of computational biology happens on this end, for example, comparison of genomes, this kind of things. And then the mindset of the people working there is much more the mindset of a genomicist or a bioinformatician. So different students, when you would tell them or would ask them where you would like to work, depending on how they feel, they would kind of choose more doing the practical lab work or more doing the kind of computational work, depending on what their interests are. The system we are working with are these water fleas. Barbara told you already, they can reproduce asexually. These are eggs from a water flea. They are identical to the mother genetically. So they are basically monozygotic clones among them and a monozygotic clone to their mother, which means that things like distinguishing between nature and nurture, or genetic and environmental effect, is peanuts in the system. It's really super easy. You can do fantastic experiments with them, and because they are small and cheap, you can do this all on a shoestring. On the other side, on the right side here, we have a Daphnia which is infected with a pathogen. It's a bacterium. The bacterium you can see up here in the upper corner here. It's called Pasteuria. And Pasteuria infects the body cavity of the host. All this orange here, this is a parasite. The parasite grows inside the host. It takes the resources away from reproduction, so the hosts are not able to reproduce anymore, meaning that they're basically completely reproductively dead, even though they're still alive. And the body becomes a shell filled with parasite transmission stages. So the parasite really manipulates the host to its own means. Has a very wide distribution. You see here all in the northern hemisphere, but then also in Africa and Ethiopia, and as Barbara mentioned, I recently collected some in South Africa. It's a model species on one side in ecology, because it's an important ecological organism. About every pond or lake you find Daphnia in there. So it's a very important, some people call it a keystone species. But then it's also a model because you can do experiments with it which you can hardly do with any other organism, and you can gain information which you then can use to understand phenomena as you would see them otherwise in humans or mice or horses. So it's a model both for the ecology of the species itself, but also a model for understanding processes which we want to apply to other organisms. So what I want to do is I want to kind of make a contrast between these two different forms of coevolution. We have here this scenario where we always get new things. New mutations replace wild types, and we have always a low diversity. While on the other side here, we have this red-green dynamics, which is highly dynamic, and it promotes diversity. So in a nutshell, you can say this one here, novelty matters, while in the red-green, diversity matters. So this was kind of my mindset when I started working on this, and we wanted to see, okay, which of the two models can better explain coevolution between Daphnia and this Pasteuria. And so we started to do experiments. The first experiment was already revealing and jaw-dropping. So really when we saw this, I thought, wow, I don't believe this. What is it? Here's a pond. We went to this pond and took a sample of Daphnia at the same place at the same moment. And then we asked, how diverse is this sample? We cloned the Daphnia, and we cloned the parasite, replicated them in the lab, and then made all possible combinations. So this is 9 × 9, 81 population. And in each cell here, the number is the percentage of animals out of 18 which got sick. So if there's a 0, it means none of the 18 animals we tested with this combination between this parasite and this host became sick. In case of 100, all 18 became sick. And it doesn't need much of imagination to see there's a lot of variation. There's a lot of variation. There was much more variation than I had ever imagined you would find in a natural population. And this is where many other people thought the same. It was just stunning. And we did this in multiple populations, trying to see is this common, and yes, it is common. We find this everywhere. So this already is very strong support for the red-green, because the red-green not only needs genetic variation to work, but also promotes the maintenance of genetic variation, while the alternative model does not. We then did breeding experiment, and the breeding experiment came exactly out like this little model here. So here we have a gene in the host, the A locus, the A gene, and you have these two genotypes here, which are resistant to this parasite or susceptible to this, and the opposite for this one here. And with this kind of scenario, you cannot get this replacement of one mutant with the other. You always keep both variants in the game. So take-home message 1 is indeed we have ample genetic variation, and this supports the main assumption of the red-green model, while on the other hand, the arms race model by directional selection with the policeman and the gangster is not supported. So we are having a scenario where we think, okay, it looks as if the red-green is really working. Now let's go back in time. Can we use this to explain what happened in the past? Let's go first on a time scale of a few months. So this is basically one summer in Switzerland. Switzerland has nice summers. You should come there. And this is a pond we were studying there. This is the prevalence of the parasite. So you see in spring, the parasite has a prevalence of 0. The parasite actually, you don't find any sick individuals. and then about mid-May, suddenly it starts, and then it goes up. At some stage, midsummer, almost everybody is infected. So this is amongst the adult animals. There are many juveniles which are not infected in this moment, and then it goes down again. At the same time, what you have here in the lower graph, this is the frequency of resistor types. So yellow, the two different types of yellow, these are susceptible hosts, and the blue one, these are resistant hosts. And you see in the beginning of the season, we have here about half of the population is susceptible, half of the population is resistant. And then this kind of stays more or less like this for 2 or 3 months, and then the parasite kicks in. And the moment the parasite really goes high, this goes drastically down here. The susceptible ones disappear. Selection in front of your eyes. Population size of this population is about 10 million. There must be millions and millions of selective deaths in this population, killed by the parasite, so that only the resistant ones are left. So evolution while you look at it. So if you thought before that evolution, these are the type of things you see when you think about T-Rex and things going far back, no. It can happen in your garden in front of you. So this is good news because the red-green model also predicts that selection must be fast and strong. Certainly this is evidence for this. Now we wanted to go further back. Another advantage of this Daphnia, this water flea system, is that in order to go through the winter, these water fleas have to produce resting eggs. Resting eggs, you can think about it something like the seed of a plant. In fall, a plant produces seeds that drop to the bottom, and then they just sit there and wait for next year. And next year it becomes warmer, more light, and then the seed produces a seedling and eventually a plant again. Same happens with water fleas. They produce these resting eggs here, these black structures. They sink into the sediments and sit there and wait for the next summer. Interesting, the parasite does the same. So this is a parasite here. These transmission stages here, they're very closely related to anthrax, and you may know that anthrax can stay decades and long periods in the soil. They can do the same. So we have now a pond, and there's constant sedimentation, and typically next year the water fleas are hatching, pick up the parasites, but occasionally a resting egg does not hatch because it's a little bit buried into the sediment or something covered it up. So they're always part of the resting eggs of the host and part of the resting transmission stages of the parasite stay in the sediment. So what you get is layers with sediments with the old ones on the bottom and the young ones on the top, and they're loaded with Daphnia resting eggs and parasite resting stages. So you can go to a pond. You have this special equipment here, which you lower into the water. It's basically a tube, which you lower into the water, and then you hammer it into the ground. When you pull it out, you have a column full of mud. And this is the old mud. And we know this mud is from 1963. And this is the new mud. This was from 2004. And it's almost linear, the time scale here. So we can now take this, we can open it, and then we have here the mud, and we can collect the Daphnia resting eggs and the parasite resting eggs out of it and bring it back to life. So we can recreate the past today. And then you can do something what, shall I say, real historians, but this means I'm not a real historian. So what most of the historians in the traditional sense of the word would love to have, we can do it, you can't. Namely, we can stop time, and we can shift time against each other. Imagine this scenario. What would have happened if this event in history would not have been the case? What would have happened with somebody else? Historians always ask these questions, kind of trying to figure out what was the significance of this depeche to this person or this attack to this place or so. We can do it. How do we do this? this is a sediment core here. You can cut it in slices. And then you can take a slice, and in the slice you have the Daphnia, the host resting eggs, and you have the parasite resting eggs. We call this contemporary, because they're from the same time slice. But then you can take the resting, the parasite spores from the next upper one. So we can take them from the future, but we can also go and take them from the past. So we have now the 3-layer, past, contemporary, future. The host is the same, so we move the parasite relative to the host. I can also keep the parasite fixed and move the host, having hosts from the future, from the contemporary and the past. So you can do this in both directions. I do it now here from the side of the host being fixed and the parasite is moving. So when you do this, you can make predictions how the infection pattern should look like. And so we did this contemporary here for many different slices. And then what you expect to find, if the red-green model holds true, then the parasites in the contemporary one should have the highest infectivity. Because later on, the population evolved again away from it already. While in the one where you are accumulating to become better and better, if you would take the gun of the lowest policeman gangster interaction, it would have worked already in the first time. It would have been better there as well. So you do get continuously better performance of the parasite over time. And so now we can use our data to see whether this or this is more the case. And the answer is the red-green model is better supported. So we have this here, and this is what we have. You see most of these curves do exactly show this kind of roof-shaped scenario. There's this one here, which is a little bit disturbing, but there we had only one single resting egg, so there was a lot of noise in the data. But otherwise, it fits very well, and you can do statistics, and you can show actually it does fit this pattern here significantly, while this one here is not supported. So this is strong support for the red-green model. This paper was elected as one of the 10 most important evolution papers by the journal Nature some years ago. So we had the process. Now we go to the pattern, because now we are describing more and more patterns rather than how things are actually coming about. Let's go back to the period about 100,000 years to 10,000 years before now. This was when most of Europe was covered by ice. Berlin is approximately here. You see, this is just maybe under the ice or maybe just tundra, but pretty cold and not very pleasant here. So, but at this time I would not have recommended you to move to Switzerland, because Switzerland was certainly under ice at this time because of the Alps. So you better should have gone to southern Spain or something. But then suddenly, 10,000 years ago, this changed drastically. Namely, the ice ages ended, the ice melted very fast. In about 1,000 years, most of the ice was gone. And Europe was suddenly a place where you can quite nicely live, and also Daphnia could move in. So the Daphnia, which had during the ice ages been hanging out somewhere here in the Middle East, they came back, and now they spread all over Europe. And we have Daphnia now all the way to northern Norway, southern Spain here. All this here is, you find Daphnia everywhere. Now, so far I showed you how the mechanism works within a population. You maintain diversity and you get the cycles. But you can also translate this to what happens between populations. So when you have different populations across space. And one of the things what you should find there is the consequence of a pattern what we call balancing selection. Namely that you find a more or less uniform distribution of the phenotypes which are important for the interaction between the host and the parasite. So we did this. This here is a map of Europe. And each triangle is one population where we collected Daphnia, and we took one Daphnia and asked, are you resistant or not to this particular parasite here, Posterior C1. And you see the red and the blue are evenly distributed. Very beautiful pattern as you would expect it under so-called balancing selection. You can imagine such a pattern for other traits which are under balancing selection. If you would fly with an airplane over Europe and would collect in every village a human being and simply ask, do you have a Y chromosome or not? Y chromosome is important in sex determination. So typically 50% of the individuals you would find would have it. And if you would then plot them in red and blue, you would get a pattern like this. It's under balancing selection. And the reason it's under balancing selection is because every offspring has a father and a mother. This is a straightforward explanation for this. Here it's because of this negative time-lagged, negative frequency-dependent selection. We did the same for the parasite. You see also here, we have unfortunately less isolates, but you see also here the blue and the red are all over distributed. Now there is an alternative. The alternative is that when here Europe became ice-free, the Daphnia were just thrown all over Europe. If this would be the case, you would also get kind of a random distribution and a homogeneous uniform distribution. But you will also see this for other traits. So we can use other traits and look into them, whether they show the same pattern. We did this. Here are a few of these traits we looked into. You see here, this is body size. And you see the Daphnia are small in the west and big in the east. This is the length of the spina. The spina is a kind of a tail's spine, which is the defensive organ against parasites, against predators. And you see they're short in the north and long in the south. This is salinity tolerance here. Daphnia are more salinity tolerant in the southwest, less so in the northeast. This reflects the distribution of brackish water ponds, which are much more common in the southwest. And finally here a trait where we don't really know what it means, but still you see quite clearly a pattern, not a random distribution, namely the color of the resting eggs, which can be totally black or largely white with only two small black spots. So clearly there's a lot of structure there, but not for traits related to host-parasite interaction. This shows that they evolve differently, and the likely explanation is negative frequency-dependent selection, this red-queen type of thing. Now I mentioned to you that this arms race between host and parasite following the red-queen model maintains genetic diversity. So let's look into those and do the same contrast here. Here we have nucleotide diversity. That's a measure for how variable DNA is. So you take a piece of DNA and you ask at how many positions do you see variation within a population? And this one here, here's a median of this histogram here. That's about 8 per 1,000. So there's per 1,000 nucleotides, you have 8. At 8 positions, there's a difference. So this one here is largely the entire genome of Daphnia. And then you see these 2 dots here, where we have about 30 per 1,000. This is where the resistant genes are. So indeed, the resistant genes are much more variable than conventional genes. How does it for the parasite? There's even more extreme. Here we have the distribution for the genetic background in the parasite, quite low genetic variation, and then there, this is where the infectivity genes are of the parasite. So indeed, this prediction of the red queen that we should have not only genetic variation at the phenotypic level, as I showed you before, but also at the genetic level is fully supported here again. Now, a long time ago, 1.5 to 7 million years ago, Daphnia magna likely occurred only in one area. This is where the species was formed. And then it spread out. And it happened to spread out over continents in a way that we have a North American clade, we have a Western Eurasian clade here, and then we have an East Asian clade. And this pattern you can very easily see in the entire genome, because this affected every gene in the entire genome. So wherever you look, you see this split into these 3 different subspecies. And now we can ask the question we said before, if the arms race maintains genetic diversity, do we see this here as well? Do we see a pattern of split between the 3 subspecies, or do we see a uniform distribution across space? And what do we see? A uniform distribution. You see here, this is over the entire northern hemisphere, plus 3 populations from Africa. And blue is resistant, red is susceptible, and you find it everywhere. You find both of them, as we had it before. And if you superimpose which of these populations belong to which subspecies, you cannot see that this here would be different from this here, or this would be different from this. Simply based on the genetic profile with regard to host-parasite interactions, these subspecies don't look different. They look the same. Now this is only one parasite. We did this for 14 parasites, and we see it everywhere. There are some differences. For example, here you see there's more red than blue, meaning more susceptibility than infectivity, and here's more blue than red, more resistance and susceptibility. But everywhere you see that in all 3 subspecies, all variation is maintained. Again, balancing selection over huge geographic and temporal space. So we are here in a time span which is about somewhere between 1.5 and 7 million years ago. Now it's becoming a bit more difficult, and those of you who are not biologists, please excuse me, but I will come back so that you will understand afterwards. We can even go further back in time. You see here we go now 15 to 70 million years back in time. Imagine you have a species, one species here, and then eventually this species speciates into 2 species. And you have now genetic diversity in the ancestral species. Then under certain conditions, this diversity can be maintained into the 2 subspecies. This is something which can only happen under balancing selection. Without this, it would be extremely unlikely that you get a pattern like what you see here, that the polymorphism you see here, you still see millions of years later in both species. If you then take these genes here and you make a gene tree, you get the gene tree which does not sort by host. You see here's host 1, host 2, and host 1, host 2. If this would be a species tree, you would have here host 1 and there host 2. So the difference between the species tree and the gene tree can tell you whether these polymorphisms were maintained since before speciation happened. These are the players. We have Daphnia magna here, and then we have 2 species which are closely related to Daphnia magna, called Daphnia similis and Daphnia sinensis. And you see the common ancestor is about 15 to 70 million years ago. This is the species tree with the 3 subspecies of Daphnia magna. So now we have, here's a species tree with blue, yellow, and green. And here we have the gene tree for 2 of 4 regions which we found, which show indeed this pattern here. And the evidence for this is that these 2 trees here, you have blue, green, and yellow in this branch here, blue, green, and yellow here, and the same on this side here. And it turned out that these 2 genes where we find this are part of the resistance complex against this parasite, which shows it's indeed that these resistant genes are maintained polymorphic with high genetic diversity for at least 15 million years. That's something which is extremely unusual. This was found only a few times, and this is the first example that this was ever found in a non-vertebrate. So people have seen this in fish and in mammals, but in insects or Daphnia or things like this has never been seen before. So, what did I tell you? I showed you that when we go over this timeline here, that we have evidence for balancing selection for diversity in host resistance against the parasite, and that there's in the parasite we have balancing selection for diversity on infectivity. And this is the solid lines here is where we have solid evidence. For both of them we have not yet waterproof, but kind of emerging evidence that it goes even further back. So possibly 140 million years back. So, take-home message 2. The red queen model has strong empirical support. It provides an adaptive explanation for extraordinary high levels of genetic diversity. And genetic diversity is a driver and a product of coevolution. Now you might say, well, these are these genes, but there's so many other genes. We know humans have about 25,000 genes. Does it really matter? Much ado about nothing. This one gene, which is responsible for resistance to Pasteuria, it actually turns out that these genes sitting in areas where they influence other genes. And for the host, we believe at least 600 genes are affected just by this interaction between Pasteuria and the host. And in the parasite, we have a massive expansion of one particular gene family, which itself, every member of the family comes in a cassette, which influences the other genes in the same cassette. So also in the parasite, we have quite a high proportion of genes which are influenced. So even so, we are talking about one phenotype, namely resistance to this parasite, about 5 to 10% of genes in the entire host and in the entire parasite are affected by this. There are not many factors which have such a big influence on a genome-wide level. Diversity matters. If diversity matters, it means that if you go to a doctor and you say, oh, I have an infectious disease, what shall I do? The doctor can't really know what he should do, because you are not the average. You're one out of the many different types of people out there. So we need personalized medicine to understand how to interact with particular diseases. That's not the case for all diseases, but for many diseases, personalized medicine is the only solution to get at a proper cure. And this is not only for humans the case, this is also for animal breeding, plant breeding, and so on. So this is everywhere where we are dealing with biological entities which are subject to interactions with parasites and pathogens. So it's important that we talk to the red queen. Coming towards the end, I want to thank all the members of my research group. The font here is proportional to the number of slides that these people have contributed to. The same here for my collaborators, and these are people and institutions which funded the work over many, many years. And if you go home today and you wonder, what did you learn? You need only to memorize 2 words. Thank you very much.